So I'm Kelly Salmon. I'm the senior scientist at the New Hampshire Academy of Science. We're a nonprofit. We operate a lab in Lyme for middle and high school students to do authentic scientific research, and we help support that at schools around the region in both New Hampshire and Vermont. Our moderator tonight is going to be Dr. Peter Fletcher, who is the executive director of the New Hampshire Academy of Science. So I've sent a message into the chat, but if you have questions during the talk, you can chat at him and he'll get back to you, or interrupt me if we need to go back. And other than that, we'll have time for questions at the end. So I'm going to share my screen to get my PowerPoint up. Oh, it's on the end slide, secrets. Okay. Whoops. There we go. Okay. So today we're going to be talking about antibodies and specifically convalescent plasma therapy and antibody testing in relation to COVID-19. As an overview, we're going to talk about the immune system in general, and then we're going to pretty quickly zoom in onto antibodies and what they are and what they do. And then we'll talk about antibody testing, particularly in relation to COVID-19, and convalescent antibody therapy, and different types of antibody therapy that could potentially be used as treatments. So your immune system is extraordinarily complicated. It's kind of amazing that we all live as long as we do and fight off as many infections as we tend to do. And this gives you an overview of the different parts of the body that are involved in immune responses. And we've written in skin here because an important part of defending your body against invaders is just having skin that protects all your more sensitive innards from everything dangerous on the outside that's trying to kill you. And there are several organs that are involved in the immune system, such as your thymus and your spleen. And people are pretty aware of lymph nodes that actually go throughout your body, in addition to the ones in your neck that tend to swell up when you're sick. But we're not going to go into great detail on any of these because you could take a whole college course plus some on the immune system and still not cover everything. A lot of what we think of in terms of the immune system is the cells that are floating around in your blood and helping defend your body against invaders. And this gives an overview of the many different types of cells in your immune system that are in your blood and in your lymph. And again, we're not going to go into all of these because we'd be here all night, but these are broadly segregated into the adaptive immune system and the innate immune system. So we're going to come back to the adaptive immune system, but let's talk a little bit first about the innate immune system. The innate immune system is able to respond quickly, but it's non-specific, and it doesn't learn from anything you've seen in the past. A response of the innate immune system would be something like when you get a cut and that gets red, it gets inflamed, and that's your innate immune system responding to something that has happened and putting out generic responses such as that inflammation to try to kill anything that might be coming in through that new entry. And parts of this may look familiar, such as histamine, and sort of the general red and unhappy is reflective of allergies as well. So your innate immune system is also involved in allergic responses. And again, that's just your innate immune system seeing something that it perceives as foreign and potentially dangerous, even if it's something that's not, like cat dander. So if we go back to all of our immune cells, so that innate immune system is several different types of cells that we see here on the bottom. And then if we go back up to our adaptive immune system, this is split largely into T cells and B cells. T cells largely kill cells that have been infected. So they're taking care of things that have already gotten into our own cells. And again, we're not going to focus in a lot on them today. But the other branch of the adaptive immune system is your B cells. And these are the cells that make antibodies and that use antibodies to basically patrol your blood and your body for a foreign invader that you've seen before. So we're really going to talk about these guys. So your adaptive immune system learns from things that you've been exposed to in the past. And it has a slower initial response because it's seeing something new. And in this graph, it's showing antibodies made on the y-axis over time on the x-axis. And it's showing that we're looking at the adaptive immune response here in terms of antibodies, that it takes about 10 days on this scale. It can be as little as 3 or 4 days to really start ramping up the amount of antibodies you're making to mount a good immune response. So it takes longer on that initial response, but after you fight off that infection, your antibodies go back down, but they don't go to zero. And we'll talk a little bit about the memory system that allows your immune system to remember that thing that you've seen before and you didn't like and you fought off. And the second time that pathogen or virus attacks you, you're able to respond much quicker and much more powerfully. So this adaptive immune system is really important to make sure that anything that we might see again, we're protected against. And the adaptive immune system is also very specific for the pathogen that it's attacking. So as I mentioned, the B cells produce these antibodies. So on the top here, we're seeing a B cell, and we'll talk more about the antibody structure in just a second. But this B cell has antibodies on its surface that are able to bind bad things like bacteria. And I should say, you have a lot of B cells circulating in you right now that are just randomly made to try to select different foreign things that you might potentially run into. If it happens to find something that is foreign that matches this B cell, that B cell gets activated through a more complicated things we don't really want to go into right now. And that allows that B cell to multiply and through a process called clonal expansion. And this makes both plasma cells, which go out and spit out tons of antibodies, which are going to be really helpful in fighting off that infection. And it also makes memory B cells that can stay in your system for months or even years that patrol to see if that invader ever comes back, and if it does, to take care of it. So what do these antibodies actually do? There are a few different options of what they can do. So the first shown here is what's called neutralization. And this just means that they're binding to the, in this case, we're going to focus on the virus one since we're talking about COVID-19. It binds to the receptors on the virus that virus needs to get into your own cells and cause an infection. And if it does this really effectively, it might prevent this virus from actually getting into your cells. In addition, having all these antibodies bound will make it so that your immune system will try to clear this virus from your blood or wherever it is, and make sure it gets cleared out of your system. One of the ways that it can do that is by a process called opsonization, which is just a fancy way of saying that it tags this pathogen and makes it look really tasty to one of the cells from your innate immune system, a macrophage, that's going to come in and gobble it up and digest that pathogen. And a third way is that activating another part of your innate immune system called complement. That is just another way that these antibodies can tag a pathogen for destruction by your immune system. And now I will also say immunologists, and I'll blame Peter for this because he's an immunologist, love fancy words. So antibodies bind to epitopes on antigens. So there are two big vocab words here that are going to come back up again. Antigen is the whole bad thing that you're trying to bind. So let's say that this whole thing is a virus. that's an antigen. It's something that your antibodies can recognize. And an epitope is a specific part of that antigen that the antibody very specifically binds to. So in this image, we have 3 different types of antibodies, the green guys, the red guys, and yellow guys, who are each binding to different epitopes, but they're all on the same antigen. So the red antibody binds to this red conical epitope, and the green antibody binds to this green, more cubey epitope. But they're all recognizing the same antigen or virus or whatever it is in your system that's foreign. And if we zoom in on these antibodies, they are somewhat complex structures, so they're actually multiple chains of proteins, and they do make this Y shape. And at the top here, on each branch of the Y, across 2 chains, you have this antigen binding fragment, and that has a specific lock and key type interaction with a specific antigen. So here it's showing it binding to this sort of turquoise antigen, but it won't be able to bind to these other ones listed down here. And this is sort of what these antibodies look like in real life. So what I'm showing on the right here is a crystal structure of an antibody. So this is how we actually look at what proteins look like in the lab. And we again see this nice Y-shaped structure. Up top, this Fab region is the part that specifically binds to your antigen or your epitope on your antigen. And this FC region down here is a constant region. And this stays the same even in antibodies that are recognizing different epitopes. And that region is what binds to your immune cells and different parts of your immune system to get recognized. So that stays constant between antibodies, but the Fab region changes depending on what you're binding. And as if this were not already complicated, there are several classes of antibodies. And we're going to focus here on IgM and IgG. There's also IgA, which is found a lot in your mucosal tissues, IgD, which no one really knows what it's doing very much, IgE, which is involved a lot in allergies. But we're going to focus on IgM because it's the first that really gets ramped up when you get an infection. So that's going to be our early stage antibody in terms of responding to something like SARS-CoV-2. And also focus on IgG because that's what sticks around in your memory, in your immune system. And it's also the most prevalent type of antibody that you have in your system. So we're going to switch and talk about antibody tests for COVID-19. Peter, have there been any questions up till now that I should address before changing topics? I will take that as a no. Okay. Also, if anyone has a question.
They have not, sorry.
Okay.
I was muted.
Oh no, okay.
There have not been any questions yet.
Okay, cool. So I'm going to switch gears and talk about antibody testing. So I put this up at the end of the presentation last week when I talked about RT-PCR based testing, because we're sort of looking at different windows in terms of the different COVID-19 tests. So this blue line here is showing the SARS-CoV-2 RNA and antigen. So as the virus is replicating in your system, this is going up, and then it's going to drop back off as your immune system fights it and defeats it and clears it from your system. The PCR-based tests can detect this infection really early on because it's picking up the actual virus itself and looking for that genomic material to determine whether you're positive or negative for the virus. As we talked about, the antibodies take a few days to really start ramping up. So IgM, this shows it becoming detectable about 8-9 days in, but it takes like a week-ish for IgM to really start ramping up to where you can pick it up in a test. And that goes up and sort of comes back down with clearing the infection. IgG goes up a little bit later and stays at a pretty good level in your memory. We're not sure exactly how long yet because we're only 6-ish months into this pandemic, so we're still figuring it out. but it could stick around for quite a while. So when we're looking at the different antibodies, if we see IgM, the patient is probably still still has an active infection. IgG, they could be well past their infection. So a lot of times this antibody test is sort of a rear view mirror into an infection that you had in the past and not a good indicator of an infection that you have right now. And something that you may have sort of surmised from talking about antibodies before is that if you have this whole virus particle rolling around in your body and you have antibodies patrolling looking for foreign invaders, it needs to be able to pick up something on the outside of this virus. It won't be able to see what's going on the inside to try to recognize that. And the most sticky outy part of the SARS-CoV-2 virus is the spike protein. And that seems to be a really good target for antibodies to recognize. And sort of the most obvious one for us to look for is for people who have an antibody response to the spike protein on SARS-CoV-2. And I'm going to talk about two main types of antibody testing that are used in the clinic. And there are other ones out there. I'm just going to focus on these two. So the first one is a test called ELISA, which stands for Enzyme-Linked Immunosorbent Assay. So the S is a little bit forced there. And what this does, and I pointed out here that this is quantitative, meaning that we can actually get an amount of antibody back from this test, not just a yes, no, but you can get what's called titers, or the amount of antibody that you have. These tests are done in a 96-well plate, like is shown on the right. And just for a sense of scale, each one of these wells, these little circles, fits about a pencil eraser. So that's the scale that we're dealing with here. On the bottom of each of these wells is AG, stands for antigen. So our antigen is stuck to the bottom of each of the wells. In this case, it's likely the spike protein because that's our most obvious antigen here. Then we add in the serum, or the part of the blood that has antibodies in it, from our patients of interest. And if the serum has antibodies that recognize that antigen, spike, then they're going to bind to it. And this binding is actually pretty strong. So we can wash away the rest of that patient's serum and wash a few times to make sure that we don't have any other contamination going on. And then add a secondary antibody. So this is an antibody that recognizes that constant region of the patient's antibody that's pretty similar from human to human. And this antibody has been made in a lab to have either some fluorescent tag on top, or in this case, it's showing you an enzyme that is able to bind to some sort of substrate that you add and make a color. And that's what's shown over here on the right, where some of these wells have turned yellow. The yellow is indicating that the, here it's talking about target protein, but in our case, it would indicate that the antibody to spike is present. And the depth of the yellow is indicative of how much of the antibody is there. So the more yellow, the more antibody. And hopefully you can sort of see in this first column, it goes in decreasing levels of yellow. And that's your reference column, that each of these contains a known amount of antibody, and you can compare all your other wells back to that column to get an approximate or a pretty detailed amount of antibody present. And luckily, we don't have to rely on our eyes for that one. There are actually fancy machines called plate readers that will go through this whole plate and assign an actual number to each well to give us, and compare that back to our knowns, to give us a titer or a concentration of antibodies that are present for each patient. So that's the more laborious, in the lab type test, but it gives us a bit more information. It gives us the titers. Then there's also a rapid antibody test. And this is a lot, the image that they show on the news is a cassette like this for a rapid antibody test. And this is just qualitative, meaning that you just get a yes or no. Do you have the antibody? And this can look for specifically IgM or IgG. or not. And the way that this works, I'll go into the real details of what's going on inside this cartridge in the next slide. But just for here, you get a little bit of blood sample, say from a finger prick. You put that into this well at the bottom of the cartridge, add a little bit of buffer, and the blood along with the buffer gets pulled along by what's called capillary action. So the same thing if you put a paper towel into a cup of water and the water goes up the paper towel. That same idea is what's pulling the blood and the buffer along this material in the middle, and that's what's going to light up in terms of telling us that we have IgM, IgG, and a control line that just tells us that the test worked. And this, or I should go through here. So there are 4 different types of results for this type of test. If you only get this control line at the top, that means that the person is negative for both IgM and IgG antibodies. But we know the test worked because this control line lit up. And we'll go into why on that in a second. If you don't get any control line, that's what they're showing you in these tiny invalids down here. If you don't have the control line, that means something's probably wrong with the test. Either the liquid didn't go all the way across, or something shifted inside the test, and you shouldn't rely on that test. You should throw it out and do it again. If you get the control line and your IgM band, that means the patient is positive for IgM, but not IgGs yet. They're probably active infection, not into memory yet. If they just have the IgG band and the control, then they're only positive for IgG, probably well post-infection or a decent amount post-infection. And if all 3 lines show up, then they have both IgM and IgG. This type of assay is called capillary lateral flow chromatographic immunoassay. And I mentioned the capillary lateral flows, that's flowing, it's capillary lateral flow across that material. Chromatographic is just that there's color involved, there's a color change in these lines. And immunoassay, you're assaying for something in your immune system using, or in this case, we were looking for antibodies. And this is the same type of test that is used for at-home pregnancy tests. So the same basic layout of the test and the same idea, you're just looking for a hormone instead of looking for antibodies. So if we actually took apart this cassette and really zoomed in on what's inside, we would find what's shown on the left here. So the sample pad is where we would add the blood and buffer. And they're showing that by just having a bunch of antibodies from the patient there. And as it starts having that capillary lateral flow across this membrane, it first crosses a conjugation pad. And living in here are 2 different types of things. So we have gold. So this is gold conjugated, meaning that we have little gold nanoparticles, which is what's going to provide the color, conjugated or stuck to COVID-19 antigen. All these tests are really quite secretive about what's actually in them because it's all very proprietary right now. I would think that this COVID-19 antigen, again, is probably spike. And also in there are, again, these gold nanoparticles for color, conjugated or stuck to rabbit IgGs. So different species, this is just going to be part of our control. And so as the antibodies from our patient flow across this conjugation pad, if they have any antibodies that bind to the spike protein, I'm just making that assumption here, that that's spike, then they're going to pick that whole complex, the spike bound to the gold nanoparticle, up and carry that with the antibody as it goes across this nitrocellulose membrane, which is just a type of membrane. And at the same time you have that whole flow going on, those rabbit antibodies are also going to get caught up in that flow and also get dragged along with all the rest of the antibodies and whatever else was in the blood sample. As they go across this membrane, they're going to hit 3 different lines. So these are the lines that we see as a readout on the test. At the IgM line, there's a line of antibodies that are adhered to that membrane that bind specifically to IgM type antibodies. And it's showing you here kind of binding along the arm of this human IgM antibody that bound to COVID-19, or the spike protein bound to our gold. And this line is actually going to capture a whole variety of IgMs that are in your blood, but only the ones that are bound to the spike, bound to the gold nanoparticles, are going to turn that color. So, you know, even if you occupied all the antibodies on this line, if there's no gold nanoparticles there, you're not going to see a color. Then the same idea at the IgG line. So this is a line of antibodies that bind specifically to IgG type antibodies. And if those are bound to spike with the gold nanoparticle, you'll get a red line there as well. Then lastly, at that control line, we have antibodies that bind to the rabbit IgG conjugated to the gold particle. And this is just proof that your flow actually went across the whole cassette, and that if, say, the liquid stopped here, you wouldn't get that control band. And you don't want to trust the test because you don't know if the liquid stopped before the IgG line. So you want to make sure that the liquid gets all the way across to this other absorption pad, and you should at least get the control line to turn red. And if IgM or IgGs that are specific for COVID-19 are there, then those will also turn red. Okay, so I'm going to switch gears again and talk about how we can use antibodies as a potential form of treatment for COVID-19. So, any questions as of now? Cool. So the idea behind using antibodies as treatment is this convalescent antibody treatment idea. So convalescent means that you've recovered from a disease. And so this is showing us here a COVID-19 patient who's now recovered. And we can remove the plasma, or the part of the blood that contains the white blood cells, including those B cells and the antibodies that are floating around. We can remove that from the patient, specifically isolate the serum, which contains the antibodies. So now we have our virus neutralizing antibodies, hopefully, because that person has fought off COVID-19. And then we can give that serum as either prophylaxis to give your immune system a boost in case you run into SARS-CoV-2. So this is particularly important for frontline medical workers, or as a therapy for patients who may be having a hard time fighting off the virus. With the idea being that this patient who successfully recovered hopefully mounted a pretty good immune response and will be able to donate some of that immune response to help someone else. And this idea is called passive immunity, because these patients at the end, both the prophylaxis and the therapy, they are getting an immune boost in a passive way. They're getting the antibodies given to them rather than them having to mount their own immune response, which would be an active immunity. So if they come in contact with the virus and have an immune response, that's active immunity. If they just get the antibodies from someone else, then it's passive immunity. This is the same idea for newborns that get antibodies from their mother. That's also passive immunity. And should have activated this earlier. This is actually what that plasma looks like isolated. So if you're used to giving blood and it turning out red because of all those red blood cells, these plasma donations are actually a nice golden color. And there are a couple of pros and cons to convalescent antibody treatment. A pro is that it's easy and rapid to access. So we now have patients who have recovered from COVID-19, and a lot of those people are willing to donate plasma. And all you need to do is get them to sit for a plasma donation. In theory, we also know this has worked for a large variety of viruses. This has been used back to the Spanish flu in 1918, and as recently as the H1N1 epidemic. And this is of relative low risk to the patients because you're just giving them antibodies from someone else. However, there are cons. So you still need to, even though you're just isolating out the serum that has the antibodies, you need to do blood testing on all the donors to make sure that that donor doesn't have something like hepatitis or another infection that they can pass on to the recipient. And it also helps if we can identify better or best donors. So this goes back to measuring titers. So that way we can pick good donors to get antibodies from. And the problem is we still need to bring in all those donors. And if we're looking at the population of the world that has had COVID-19 compared to the population that has not had it, there's a big difference there. So it's not a very large scale treatment, but it's a good treatment for right now, especially for patients who are having a particularly hard time fighting off the virus. So a step better than getting these convalescent antibodies from people is actually manufacturing antibodies. And we can actually do this in huge tanks like this one. And this is already in place for a lot of prescription drugs. So things like Humira are actually monoclonal, we'll get into that in a second, antibody drugs. Oh, we're getting into it right here. So it's an important distinction for manufacturing antibodies to distinguish between more vocabulary, monoclonal versus polyclonal. So if we look at all three, these red, green, and yellow dudes, if we take all of them together, that's polyclonal. So there are 3 different types of antibodies, though they are all recognizing the same antigen. But we have variety in what specific parts of the antigen that they're recognizing, and the antibodies themselves are different. If we just look at one of these types, say these green guys, and we only have a bunch of those, that's monoclonal. They're all the same, so one clone. And there is some manufacturing going on and larger scale treatments that are in production for being on the polyclonal front. So there are a few efforts to better purify human serum, so that way we can better get antibodies out of people who already have had COVID-19 and get that distributed to more people. Another example that I just thought was interesting is from a company, SAB Biotherapeutics, which is immunizing humanized, and in this case, the humanized, it's only that they're expressing human-looking antibodies. They're not like half-human cows, but humanized just in the antibodies cows, and giving them the SARS-CoV-2 virus so that the immune system will mount a response, and then collecting these human-like antibodies from the cows, which produce a lot of antibodies, potentially for human treatment. And my personal favorite, there is also work being done in llama antibodies. So this is Winter, who is a llama who was previously, actually before the pandemic, had been given the SARS virus and the MERS virus, which are very closely related to SARS-CoV-2. And they were using this llama to produce antibodies against SARS and MERS that could be potentially used for human treatment or for research. And once this pandemic hit, they started deciding to see if the antibodies that Winter is producing could potentially also pick up SARS-CoV-2, which it turns out some of them could. And llama antibodies are particularly interesting because in alpacas and llamas, they have some antibodies that look like ours that have those multiple chains, really big honking things in terms of proteins. And you're binding your antigen along these 2 different chains here and over here. And alpacas and llamas have much smaller antibodies as well that only have one region that binds to the antigen. And actually you can even just snip off this bit, and that's called a nanobody. That it's just this one little piece of antibody that is still able to go and bind and potentially neutralize, if we're thinking about neutralizing a virus, without having to have this huge thing that you're putting into your system. And this is potentially also a lot easier in terms of production, and especially these small nanobodies can, once they're made, or once we know what they look like, we can more easily make them in the lab, as well as not having to have a huge army of llamas, even though I would personally be a fan of a large army of llamas, to make all these for a potential human therapy. And then if we switch to thinking about monoclonal antibodies, then we're just making a whole bunch of one type of antibody. And this is actually a lot easier in terms of manufacturing, even though I'm first going to put up a slightly more complicated chat to explain that. Whoops. Or sorry, I pulled up my chat at the same time because I saw something. But put up a more complicated image to explain this at first. So this is using a mouse as an example. And for some reason, in this case, the mouse is being immunized against sheep red blood cells. Don't ask me why. But we inject the mouse with the sheep red blood cells. The mouse makes an immune response against it. We can then isolate the mouse's spleen, sorry mouse, and from that get a bunch of antibody-producing spleen cells. And these might be making different types of antibodies, which are shown in the different colors here. Then we can force those cells to merge or to smoosh with myeloma cells, so a type of blood cancer cell. And the reason that we do this is that if we can effectively squish those together and make them one monster cell, the myeloma is going to make this antibody-producing spleen cell that we want immortal, so that it will just keep living in culture, which cancer is very good at. And those cells will just keep pumping out unlimited amounts of antibodies. And when we make this fusion, it's then called a hybridoma. And then we can grow up those hybridomas. And in this case, they're showing that you're now making monoclonal antibodies specific for sheep red blood cells. But we can keep growing these up pretty much infinitely, and then grow them in huge bioreactors like this one, and they get even bigger. This was just a good picture I could find that had humans for scale. And if you keep giving them food and give the media that they like to live in, they will just keep pumping out antibodies. You can suck the antibodies out of this big bioreactor and give them more food, and they'll just keep making more antibodies. So this is a really scalable way of making a ton of antibody. And there's already been discussion. These bioreactors are also very similar to brewing tanks. So if we get a good line of monoclonal antibodies that look like a really good therapy or even prophylaxis for humans, there's discussion about taking over places like Anheuser-Busch that have huge brewing tanks as sites that we can manufacture tons and tons of antibodies. So to kind of look back, I know we covered a lot tonight. There are some really good potentials in terms of treatment. So the one that's really on the market right now, being used in the clinic, is that convalescent antibody treatment. And then sort of more forward-looking that are in production or trying to figure out what works best. There's a couple of different polyclonal antibody solutions in terms of either raising polyclonal antibodies in other animals or isolating them better from humans, and also monoclonal antibody production. And this can be used as both a treatment for patients who already have COVID-19 or potentially as a prophylactic to protect people who might be exposed. But what we're really going to need, and hopefully going to get at some point soon, is a vaccine to get the herd immunity that we're going to need to really beat this back. And that's what we're going to talk about next week. So next week is going to be sort of a historical look at vaccines, and then what's going on right now in the vaccine world for COVID-19, which is actually some completely new and different ways of making vaccines than anything we've done before. And with that, I'd like to thank again the Claremont Makerspace for allowing us to do this collaboration and making this hosted on their website. And again, I'm with the New Hampshire Academy of Science. Our website is here, and here's our Gmail if you want to contact us about anything. And we do outreach like this, as well as a lot of programs aimed at middle and high school students to actually do real research, and also programs for middle and high school teachers. And we also have a satellite lab at the Fairbanks Museum and Planetarium. And with that, I will open it up for questions.